Okay guys, so let's just quickly review ecosystems, section 5.1 from the textbook. It's all about ecosystems, and let's just quickly discuss the general structure of the topic. Essentially, topic 5 is really about climate change, building on some ideas from topic 4. So it begins with an understanding of what ecosystems actually are. It moves on to talk about abundance and distribution. So in an ecosystem, we've got lots of different organisms, but what are the factors that affect where in the ecosystem these organisms are? So what factors influence their abundance and distribution within the ecosystem? Because if we look around in an ecosystem, we don't see all the organisms everywhere. So just look in this picture, for example, if we have this ecosystem right here, we're going to get all the plant life forms, all the plant organisms, like trees and all the land-based organisms. They have adaptations to survive on the land here, whereas we might have aquatic organisms with aquatic adaptations in there. So even though we have a whole ecosystem right here, what affects the distribution of the organisms? Because the fish won't be on the land, the trees won't be under the water. So what is it that affects where those organisms are and how many of them there are there? So that's what comes in the first part of this topic, section 5.1, is what are the factors that affect abundance and distribution. And that's what we will look at, not only what those factors are, but how you can measure it. So how you can measure the effect of those factors is a technique that we call ecological sampling, and that's essentially the core practical that many of you did at the end of the term. So that's ecological sampling, looking at the factors that affect abundance and distribution. The next section that we'll look at is climate change. So that's what you'll be doing over the summer, or what you did over the summer, and that's looking at global warming and the evidence for global warming and how to interpret that evidence. And then the last section really is about looking at climate change and how the impact. So what is the biological impact of climate change? So let's do it. So the first thing we looked at was the definitions of the key terms that we'll be using throughout the topic. Obviously there's other ones, but the key ones are ecosystem, habitat, community, population, and niche. Okay? And we looked at this idea that within an ecosystem, the key ideas here are that the ecosystem is all the organisms, which is basically the living component, plus the habitat, which we can think, or the environment, which we can think as non-living. Okay, so it's basically everything, but that everything, we must understand, comprises of the living organisms plus the non-living component. The particular place is the habitat, and this is not always strictly true because the habitat of some organisms is another living organism. However, we can think of the habitat as the environment, okay, that the organisms live, and more or less we can think of that as the non-living part. The community we can think of in general terms as all the organisms, so all the organisms, including all the different species interacting together. That's the community, and the population is what the community is made up of, many different populations. So many different populations make up the community. The community plus the habitat makes up the ecosystem. So you can think of it like that. Okay, let's move on. So we had that little exercise on looking at any given example and being able to describe from that example what makes up the population, what makes up the community, what's an example of a habitat, and what's an example of the ecosystem. And you should be able to do that for, any given scenario. Okay, so we will move on. So then we talked about this key question about what an ecosystem is. And it's just to get you to appreciate why the ecosystem looks the way it does, what are the characteristics of different ecosystems and why. Okay? So we talked about this idea that, this is to answer this basic first question. So why do we have ecosystems? Another way of asking that question is why are there different types of organisms in an ecosystem? So why are there more than one different types of species? Okay. And then we come across this idea that, well, every single living organism has to have energy, right? And needs to have a source of energy. You can't extract that energy itself from nothing. Yeah, it needs some kind of energy input. Okay, and they need to be able to survive. So the two things that organisms need are energy and being able to survive so that they can reproduce. Those are the two things that they need. And what we're going to do is use these two ideas to explain why there's different types of organisms. Long story short, different organisms have adapted different ways of getting these two things. Okay, they've adapted different ways to obtain and extract energy. They've obtained different ways or adapted different ways to survive and therefore reproduce. So let's begin then. So we have this idea that every organism is made of cells, and every cell, in order to function, it needs ATP. And in order for that cell to be able to make ATP, that cell needs an input of what we call complex organic molecules. Complex organic molecules. Because complex organic molecules, being broken down to simple inorganic molecules, is what releases the energy that we can use to make ATP. And ATP is a little kind of battery molecule that the cell can use to get energy in little packets that it needs to do little things. Okay? So, in order to convert complex organic molecules into simple or inorganic molecules, and remember, this is basically what we call the process of cellular respiration. In order for cells to be able to carry out respiration, they need a source, therefore, of complex organic molecules. Okay? And this is the idea. This is how energy gets into ecosystems. All right, so we say every organism needs a way to have these complex organic molecules. But this is our then first type of this is our first time that we look at how different organisms have adapted differently. Some organisms have adapted to be able to generate their own complex organic molecules, and they make these complex organic molecules using energy from their environment. And species that can do this are called autotrophs. So they are making their complex organic molecules themselves, and once they've made those complex organic molecules, they break them down in the process of respiration, releasing the ATP, and then allowing the cell to carry out all the things that cells need to do, including grow and form tissues and organs and so on. Okay, so that's autotrophs. Heterotrophs, on the other hand, cannot synthesize their own complex organic molecules. So, for example, let's just rewind a little bit. Autotrophs via photosynthesis, so autotrophs convert simple inorganic molecules into complex organic molecules. Okay, so autotrophs can convert simple inorganic molecules, carbon dioxide, water, into complex organic molecules such as glucose, such as starch, and so on. Okay, but the process by which they do that is called photosynthesis, at least the plant. So the photosynthetic organisms carry out photosynthesis. There's other types of organisms that do this in other ways that we can't call photosynthesis. However, by and large, it's photosynthesis. Okay, so they create their complex organic molecules for themselves, and then they break those down in respiration. Okay, producing ATP and converting the complex organic molecules back into simple inorganic molecules. So here we have then two different types of organisms. They've adapted different ways of getting these complex organic molecules. We call them autotrophs, and the ones that can't get those molecules or can't make those molecules themselves, we call them heterotrophs, and they simply consume the plant material, or they consume the producer. So the producer is an autotroph. The producer uses some external form of energy to make complex organic molecules, and they use that for growth, whereas the heterotrophs consume those pre-made molecules without having to make them themselves. But both of those organisms need to carry out respiration to make ATP. So the next stage in the idea is, well, okay, so we've got our producer right here. Why are there different forms of these consumers? Well, the simple idea is that different species have adapted different ways to obtain their complex organic molecules. The grasshopper is adapted to eat the plant. The bird is adapted to eat the grasshopper. The snake is adapted to eat the bird. The owl is adapted to eat the snake, and so on. So because all these organisms have adapted different ways of getting these complex organic molecules, this is why we have so many different organisms, because they've all adapted to find their own niche. Okay, so they've occupied a niche. So at some point, there might have been lots of energy accumulating, in all the snakes. However, with the evolution of certain adaptations, this species was able to take advantage of all the energy that was building up in the population of snakes. So the owls now had access to all that energy. So the owls evolved to occupy the niche, which allowed them to get energy from the snakes. Okay? So the ideas of adaptation and niche that we encountered in topic 4 kind of are built upon next. Okay? So one reason, therefore, why we have all these different organisms in the ecosystem is because they have had to adapt differently to ensure that they have a source of energy. Next, adaptation to the environment. And this is really a lot about survival. And obviously getting enough energy is a part of that, but so is being able to adapt to your non-living part of the environment. So the idea is that different ecosystems have different environments. Yeah? Different ecosystems have different non-living conditions. And those non-living conditions you can think of as acting as a selection pressure. act as selection pressure. And because that selection pressure exists, organisms adapt in different ways to survive. Or those that adapt survive, and once they survive, they form populations within an ecosystem. So if you see any group of organisms present in an ecosystem, the only reason why they are there is because they have developed the specific adaptations to survive those conditions. Okay? So, because different organisms, different species have adapted different ways to survive given the same conditions, that's another reason why we have so many different types of organisms in the ecosystem. Okay, let's move on. Then we had a review of all the terms, and then we started looking at the factors that affect abundance and distribution of organisms in an ecosystem. Okay, so the next thing then is factors affecting the abundance and distribution. As we discussed before, the introduction was about what makes an ecosystem up, and the next thing, and this is the major thing in the first section, 5.1, is looking at the factors that affect where these organisms are, i.e. the distribution. So the distribution is really where these organisms are, and the abundance is how many or how much of that organism is there. Okay, so that's generally what those terms mean, and then we look at the factors. So the first key factor is, number one on that list is the abiotic, biotic, and anthropogenic factors of that ecosystem. What does that mean? Abiotic is the non-living conditions of that environment. The biotic obviously means all the kind of factors which depend on living organisms. Okay, so you can think of it as living factors. And anthropogenic really can be either of these two, which are heavily influenced by humans. Okay, so for example, we could have grazing. Okay, now grazing is a living factor because it's basically when you have some herbivores who are munching, are consuming the producer, and that affects the growth of the producer. So that's a living factor affecting the abundance and distribution of grass, for example. Okay, however, if those organisms are grazing there because it's a farm area or it's farmland that's kind of maintained by humans, then that factor is anthropogenic. That factor then becomes anthropogenic, even though essentially it is a biotic factor. And the same applies to abiotic factors. So something could be an abiotic factor, for example, temperature, but if that temperature is affected by humans, then that then counts as an anthropogenic factor, not just an abiotic factor. Okay, so these are the factors. And then we looked at some examples. So, for example, lichens are a kind of mutualistic relationship between a fungus and an algae or bacteria. Okay, and then if we look at this, the distribution here. So what we're saying is that the distribution of the lichen is affected by the rainfall and the slope of the roof that makes some parts of the roof have more water than, or have more water availability or retention than in other places. So what we're saying is that the abiotic conditions in this case are affecting the, where the lichen can grow more. So we're saying that this is an example of an abiotic factor. So this is what we need to consider. So when we look at other things, like let's say, we have some tiles like this on the ground. So it says this is someone's driveway. And what we see is usually the plants that first colonize this area, why don't they grow on top of the tile? So, how come they're only growing in the middle gaps, in these gaps right here? How come usually you don't see these plants, mosses, weeds, or whatever you want to call them, how come they're not growing from the middle of the tile? That's because there's certain factors which influence where these plants have a better chance of survival. Yeah? So in between the tiles, maybe dust and soil and mineral accumulates, which then makes the basis of some kind of soil that these plants can access, which can retain water and then can allow their growth. Whereas the tile, with its surface, doesn't allow the accumulation of those nutrients or water, and therefore the plant can't grow there. Okay? So what we're saying is that the distribution of organisms around us is not random. It is affected by certain factors, and we need to be aware of those factors. And furthermore, now we should be able to label those factors as biotic, abiotic, or anthropogenic. Okay? So then we did a bit of observation. We looked at what those factors are, and you need to memorize those, and you need to have a couple of sentences in your mind ready to say how those factors can affect the abundance and distribution. So for example, solar energy input. How could that be a factor? So I've memorized it, I know it's a factor, but how could I explain that? So I need to think. Solar energy probably affects photosynthesis, and how much photosynthesis can happen. How much photosynthesis can happen then affects how much energy is moving through the ecosystem, and the more the energy, the greater the abundance is likely to be. Okay? And in terms of distribution, it's like wherever there's more solar energy input, that's where the abundance is going to be higher. Okay? So remember, abundance is all about how much. Distribution is when you start talking about different places, different locations in which there's higher or lower abundance. Okay? So then we looked at some examples, and we tried to think of the reasons why certain species were more abundant in one area versus another area. Yeah, and we talked about, it might be that, this area might be watered, for example. This area might have less competition because the grass is mown. Yeah, so you've got to think about all those factors. That's just applying what we've looked at. More examples there, and then we went through the review questions. Organisms are adapted to a specific set of biotic and abiotic conditions. All right. So remember what we said at the beginning. Organisms are adapted to the particular conditions of that ecosystem. Okay. So if they're adapted to a specific set of conditions, and you start changing those conditions, obviously you're going to affect the abundance of those species. Okay? So that's the whole premise of the first section. Presented by an ecosystem. Okay? The way they exploit these conditions, it's called its niche. Clue was that. Okay? The abundance refers to how many of a particular population, species. The distribution refers to the location, or where. Okay? As each species has adaptations to survive within a particular range of conditions, their abundance and distribution is affected by those conditions. Okay? And we call those conditions the biotic, abiotic, and anthropogenic factors, right? And the examples we've already been through. Okay, so gave you some space there to kind of think of your own reasons why these factors would affect the abundance of any given population. And then we looked at the second thing. So the second thing that affects the abundance and distribution of organisms, we've already kind of looked at it or mentioned it. It is adaptation. Okay, adaptation. So organisms find themselves in a particular place. If they can survive, it's only because they have the adaptations to do so. So as well as the biotic and abiotic conditions favoring their adaptations determine the places where they can survive. Okay, so that's a bit more straightforward. Okay, and we looked at the example of the moths and we looked at malaria, but the general idea is that organisms will have a higher abundance, or populations will have a higher abundance in the places which have the conditions should have thought this through, I think. Organisms will have a higher abundance in the places which have conditions, and we're talking about the biotic and abiotic factors here, which have the conditions that their adaptations allow them to survive. Okay? So, you have a polar bear, for example, and the polar bear has got adaptations to a particular area, particular set of abiotic and biotic conditions. You take that polar bear somewhere else with a different set of conditions, that population of polar bears will not be able to survive there, so their abundance will go down. Okay? And, I mean, we can use this example. So here, what we've said is that we have malaria. Okay? It's adapted to around about 20 degrees C. So that's 20 degrees C is like the minimum temperature. Okay, and what we're saying is that if climate change happens, then it's going to start to occupy different areas. Okay, so climate change increases the temperature in places that were below 20 degrees C. Okay, so now malaria, if climate change happens, malaria can survive in new environments. And we're talking about countries. Okay? So the malaria can now survive in new environments, and therefore abundance of malaria will increase in those new countries or different countries. Okay, so what we're saying is that if global warming increases, it increases potentially the number of countries that have a temperature higher than 20. So therefore, malaria is no longer limited to the yellow distribution shown on the map. It may then start to occupy the red areas if climate change continues, because its adaptation is allowing it to survive in those places, which now, so the abiotic condition of those countries have changed. Okay, so if we take this area right here, for example, what we're saying is currently there's no malaria there. But if global warming continues to happen, then the temperature of this country might increase to 20 degrees C. And if that does happen, then the abundance of malaria will increase there. Okay, so we have to review. And last but not least, the third and final thing that affects the abundance and distribution of species is succession. Okay, so we discussed this in lesson that succession, there's different types, but essentially succession is a process whereby the community changes over time in a particular area. Okay, and this is its basic definition. Now there's more specific types of succession that we need to be aware of, but essentially the community changes over time in a particular area, and the basic concept here is that populations have the capacity to change. It's not all populations, but in succession what happens is a population changes, populations change abiotic. Populations are responsible for changing the abiotic conditions. As abiotic conditions change, we've already discussed, as abiotic conditions change, obviously the populations that can survive there also change. So by species A being there, species A changes the conditions. Species A changes the conditions, which means that species A can no longer survive there. However, species B can survive there, so species B replaces species A. Then species B starts to change the conditions, makes it more colonizable by another species, species C, and in doing so, species B can no longer survive there, whereas species C can. So this is the process of succession, all right? And it's really specifically all about the communities, all right? It's about the living organisms in that place. So let's look at that process, okay? So what we have is the pioneer species. And what we're saying is that, at least in terms of primary succession, primary succession is when we're talking about the change in a community over time in an area where there was no communities, no species of life in the beginning, which is basically every place. Every location on the planet at some point was lifeless. Okay? So, when we talk about a lifeless place, developing communities of living organisms, that process is called primary succession. Okay? And primary succession begins with an understanding that an area that is lifeless because it's got extreme changeable conditions. Remember this. And better, it's got extremes and very changeable abiotic conditions. The more ideas you can link together, remember, better chance you have of collecting marks. So, lifeless places have very extreme or changeable abiotic conditions, and this is difficult. This is difficult for many organisms to adapt to. Or another way to think about it is that not many organisms have the adaptations it would take to survive these conditions that are, at one part of the day one thing and at the other part of the day very, very different. Okay, most organisms have adaptations to a specific set of constant conditions. Okay, so these abiotic factors are difficult to survive, and that's why they are lifeless. However, some organisms do have the adaptations to survive these places, and they are called pioneer species. Okay, these are their characteristics, which explains like why they can occupy certain places. So these are called pioneer species, right? So they can, whereas some other plants would need a lot of soil with a high organic matter content, they might need soil which is very moist, they might need soil with high levels of minerals. Other plants, can survive basically where there's any collection of dirt, okay, or a bit of sand. So these species we call pioneer species. Now what the pioneer species does is it changes those abiotic conditions by its presence. So for example, in this area, what would happen is that the grass that you might see, or don't want to call them weeds because weeds is a relative term, but let's just say that whatever this pioneer species is, grasses, mosses, even these lichens, what they do is as they grow and die and as part of their normal life cycle, it results in an accumulation of organic matter. Okay, and with the accumulation of organic matter, it's also able to retain water. So the presence of the grass, mosses, and lichens, these pioneer species, as over time, it results in accumulation of organic matter. It results in a bit more of kind of the ability to keep water, and I hope you can see that naturally this is resulting in a change in the original conditions. Because before the grasses and mosses and lichens came, the organic matter content of this habitat was practically zero. The ability of it to retain water was not very good. But the presence of the grass, moss, and lichens has changed that. And what that does is it has changed the abiotic conditions. And now we might have abiotic conditions that some other species are adapted to, whereas they weren't previously. Okay? So before, it's not that the adaptations of other species are changing. What we're saying was that the abiotic conditions of this place didn't suit any organism's adaptations. But now, because the abiotic conditions of this place have changed, now we might find that there are some species that are adapted to this place. Okay? And that's what happens. Yeah, that's what happens, is that the pioneer species kind of changes the conditions until you get a little bit of soil. And then you start to get mosses and grasses and a bit more taller plants, and they come in. And as the soil, and so because of their roots, the roots bind the soil and further improve the water retention, because otherwise the water just goes through, right? But with the presence of the roots that binds the soil together, you get a bit better water retention, and that improves the soil further. And as the soil quality improves, larger, taller plants colonize, and they outcompete the pre-existing ones. So as this place's abiotic conditions are improved by certain species, the next set of species comes in and outcompetes the previous ones. So these pioneer species, over time, they are lost from this place as they're outcompeted by the taller plants which are able to outcompete them for light. Yeah, so because they're taller, obviously they'll outcompete the shorter plants for light, and the abundance of those other pioneer species will go down over time. The same process continues. So these taller plants are themselves responsible for changing and improving the soil content even more as they live and they die. And as they do that, they improve the soil quality even further and allow even taller and other plant species that have a higher demand for water, have a higher demand for organic content and nutrients from the soil, but those plants happen to be taller still, and they outcompete the first set of tall plants that came into the area. And this process continues until you reach a stage called Climax Community, where you got tall trees that are the dominant species, and no other species can, in fact, come in at this point to outcompete that population. So at this point, you get the process of succession effectively stops. And this is called the Climax Community. Okay? And this diagram right here is very important because it allows you to see in one kind of instant what the process of succession would have been in this particular location that now has trees. Okay, so what they're saying is if you came back like 1,000 years ago, you might have seen this population in this area. If you came back 2,000 years ago, you might have seen these guys in this particular area. So looking at what the change, looking at the distribution of plant species in this direction in space allows you to kind of get an idea of the process of succession which happened in one location over time. That's right. Okay, that was basically that, and then we kind of looked at some other examples of succession where there's variations. So in some cases where the soil quality is very, very poor, the climax community is not trees because the soil quality is so poor it can't support those large organisms. And so it's just kind of giving you the idea that climax communities aren't always trees and forests. We've got secondary succession, which is effectively, it's basically restarting the whole process. So we've had a climax community here, but there's been some kind of natural disaster, and we've lost, let's say we lost all the community. Well, what happens is that it doesn't start from scratch. It doesn't start from all the way at the beginning, which is a lifeless place, because even if we lose all these communities of organisms, let's say we lost them because of a fire, it's, the soil is still high quality. So we don't have to start all the way at the beginning where we need to form the soil and we have a completely lifeless area. Okay, we probably have like seeds and pollen that's blown, seeds and other things that have kind of blown in from other areas, and it's ready to go because the soil is already there and it doesn't need to be developed. So secondary succession is slightly different to primary succession. Okay, and then we've got deflected succession, which means, this grass was going to grow tall. This grass was going to grow quite tall, and then, as the grass grew tall and it changed the conditions, and then taller plants could come in, and then taller plants could come in, and then eventually we would have trees. But we're not going to have that now because these grazing organisms are kind of keeping the grass really short, which means that the tall plants never really get to come in and outcompete the grass. Okay, so, or if they came in, they would just get munched immediately. So grazing effectively stops the process of succession in its tracks and keeps it at a particular stage. All right, and it never moves beyond this stage. So this is called a deflected succession. Okay, and then we had the activities, and that, I think, is that. ecological sampling is really just another way of saying how we can investigate the effect of various factors on the abundance and distribution of organisms in an ecosystem. Okay? So in the first part we said we went through the things that could affect the abundance and distribution of organisms, abiotic factors, adaptations, and succession. Ecological sampling is how we would practically investigate the effect of these factors on the abundance and distribution. So it's the kind of practical side of that. The first key point, I mean, there's a number of slides here. I don't want you to ignore any part of this, but the basic idea of sampling is this, that you have a large area and you want to be able to do an investigation and apply a general rule or relationship. So you want to understand, for example, the effect of light intensity. You want to apply it to a large area. However, you probably don't have the time to investigate and count the abundance and distribution of species in that entire area. So what you do in place of that is to sample a small but representative area and then apply the findings that you got from that area to the larger area. That is what we call sampling. Okay, and in order for sampling to be, in order for it to have any kind of usefulness, the sample has to be representative. Okay, the sample has to be representative, i.e., measuring one quadrat from this area might not give me the full picture. Okay, so if, let's say, I just draw some dots here. So where there's dots, let's just say that there's a particular species in this area, and I want an idea of how abundant that species is. If I took one quadrat, 50 centimeters by 50 centimeters, I could get some data from that quadrat and multiply that up by whatever this number is, that multiplied by that. I could multiply that up, and whatever results I get from this one single quadrat, I could multiply that up by the factor and estimate what I would expect to see in the larger area. However, if in that quadrat I didn't catch any of this particular species, it would tell me that I would then extrapolate that there were none of that species present in this area, which would be inaccurate. It would not be representative. So my conclusions would not reflect that particular area. So there's probably a certain number of times that I would need to repeat this quadrat randomly until I got or recorded some daisies. Okay, so the number of quadrat it takes in order to get a representative sample is one consideration. So number of samples, or in other words, sample size. So sample size is a big factor in if that sample is representative or not. The other one is being unbiased. Okay, and one of the ways that you can be unbiased is by doing random sampling. Okay, so you do not choose yourself where you're going to put the quadrat. You let some randomly generated values tell you where you're going to put the quadrat. The other way to do an unbiased sample is to be systematic about it. Okay, so let's say in one area I've done a random sample. In the other area I might do a systematic sample. So maybe something like this. Shaded boxes represent where I'm going to be sampling. Okay, that might be a bit overkill, but that you have to decide. Oops, I lost my pattern there. Anyway, you get the idea. All right, so the idea of a representative sample is very important. Now, we come to quadrats. So our core practical was effectively looking at plant species. Now remember that whenever you're doing sampling, you're looking in any investigation, whether it's ecological sampling or not. Remember, our golden rule was that we are looking at something, the effect of what we call independent variable on what we call dependent variable. Okay? For example, the effect of substrate concentration on enzyme rate. Dependent variable, independent variable. And in the same way, when we do ecological sampling, we must be looking at those factors as well. But there are some differences. Okay, so in an ecological sample, we're looking at the effect of some abiotic factor on abundance and or distribution. So always remember what it is that you're measuring, what it is that you're looking for a correlation between or a difference between. Okay? So the effect of an abiotic factor on the abundance and distribution, this is our independent variable, this is our dependent variable. But the way we have to measure these things is slightly different in an ecological sample versus, for example, a laboratory investigation. Okay? But anyway, we discussed about how to use the quadrat there. So one way that you can make so let's just talk about the two different ways that we can do investigation. So the two ways that we can set up an investigation is each time you do a sample, let's just go back to this, each time you do a sample, you need to record two things. One, record your abiotic factor for each quadrat. So for each place that you're recording or sampling, you must record the abiotic factor. This is our independent variable. If we didn't record it, we don't know what influence it potentially could be having. Two, we must record. So we record the abiotic factor and we record the species abundance. Remember, sometimes you might be specifically looking for the abundance of daisies, a particular plant species. Okay, in that case, you're only recording however many daisies you find in your quadrat. But in other cases, you might be recording all the plant species that are present and looking for particular relationships within that. Okay, so you must record the abiotic factor that you're interested in. You must record the abundance and distribution. And three, you must, if possible, record other factors, other abiotic factors, any other abiotic factors that may influence the abundance and distribution. So anything else that you think could affect the abundance and distribution, if it's within your means, you must record those other factors, because you don't know whether it's one of these factors or the factor that you think is influencing the abundance and distribution that actually is affecting the abundance and distribution. Okay? And remember, this fits in with our three practical considerations that we've been sticking with all this time. We have to say what is our independent variable, we must say what our dependent variable is, and we also must give consideration to our control variables. So these are the ideas here that we're slightly modifying because it's an ecological study. And remember, in a laboratory investigation, you can control the control variables, you can change the independent variable, but in an ecological sampling study, you are not in control of those things. Okay, so for 3, the only thing I would add to that is either you record other factors or you set up your investigation so that these other factors can't influence the thing that you're looking at. Okay, so now that we've decided like how we are going to record the data, how we're going to, or what is the important information that we're going to record, we need to think about how we're going to set up the investigation. And there's 2 general ways that you can do this. The first is a simple area comparison. So there's something different about an abiotic factor in area 1 and area 2. So something is different between the 2 areas. So then I take area 1 and area 2. For obvious reasons, the size of the 2 areas should be approximate. The number of quadrats I take in each area should be the same. So these squares represent a different quadrat being sampled. Okay, and I take a certain number of samples. So the number of samples has to be representative. Number of quadrats representative. Okay, it has to be random in each area, and in each area I will record the 3 things that we discussed. Okay, so I'll just put down 1, 2, and 3 here, referring to what we discussed before. So, okay, independent variable, the abiotic factor that I think could be influencing things, my dependent variable, the abundance of the different species within each quadrat, and 3, any other factors that I think could be related or as just kind of control variables. I just want to make sure that they're not different in these 2 areas. But I take these measurements for both the areas, for each quadrat, and I put it into a table. And the table, approximately should be like, so here I've got quadrats. So quadrat 1, quadrat 2, quadrat 3, quadrat 4, quadrat 5, and so on. And down here, I've got the species. So species 1, species 2, species 3. And in these boxes, I'm putting the percentage abundance. Okay, and it doesn't have to add up to 100. Whichever, each square that the species is present in, it gets counted as occupying that square completely. And so we have that, and then if you're doing an area 1 versus area 2, you're going to have this for area 1, and you're going to calculate averages of each of the things across all the quadrats. Okay? And then, so you're going to have an average of all the things from area 1, and then on a separate table, you're going to have averages for all these same things for area 2, average area 2. And then you do your investigation. The analysis is by comparing the average of here, so the average of species 1 in area 1 versus the average of species 1 in area 2. And then you see whether there's any significant difference between the 2. And ideally, you test that significant difference with a t-test. But more on that later. Statistical test for different means. Okay, so we do a t-test to see if the average of species 1 in area 1 is actually different from the average of species 1 in area 2. And we repeat these for the other things that we want to compare. And hopefully, well, not hopefully, but if there is an influence of a particular abiotic factor, there's significant difference between abiotic factor and the species abundance across both areas. So that is what we call area quadrats. Okay, so comparing one area versus another. The other way you could set out your investigation is by a simple linear pattern of quadrats. So let's say you think that from point A to point B, you think that the pH is changing. Okay, so what you do then is, and you want to investigate the effect of pH on species abundance and distribution. So what you can do is take quadrats not in a two-dimensional area, but in a series that goes across. And again, you take the recordings of 1, 2, and 3 in each quadrat, but this time you go in a line. Okay, so in this case, your results are going to be quadrat 1, quadrat 2, quadrat 3, quadrat 4, quadrat 5, and you're recording your species 1, species 2, species 3, and your pH. And maybe as a control, you're also measuring light intensity, making sure that that's not changing. And when you do this, the way you analyze this data is you see whether there's a correlation, not a statistical difference, but you see whether there's a correlation between the abundance of species 3, for example, and the changes in pH. Or maybe the change in pH going this way is more correlated with the abundance of species 2. So species 2 is more sensitive to pH changes than species 3, for example. So when you analyze this kind of data of transects, what we do is the distribution is not random. So when you take your quadrats, it has to be systematic at regular intervals, not random. Okay? And when you analyze the data, you're testing for correlation, not difference. And the way you do that test is use Spearman rank correlation test. Okay, so those are the two kind of ways you can set your investigation up, and it really depends what factor you're investigating or what type of area you have, which will determine which kind of study that you do. Okay, so that is ecological sampling. The slides go on to discuss about kind of the finer points about how we plan the investigation, but I've just discussed the kind of key components there with you. And that brings us to the end of section 5.1. Section 5.2 is about energy in the ecosystem. How does energy get into the ecosystem and how does it move through the organisms in that ecosystem?